Good morning. My name is Chris Chumsey. I'm here with my co-moderator, Darren, and it's my pleasure to welcome you to Day 3 of the CAS Mass Spec Conference 2021. It's with great pleasure that I have the opportunity to introduce Professor Brandon Rutlow. Professor Rutlow is currently a professor in the Department of Chemistry at the University of Michigan. Professor Rutlow received his BS in chemistry from St. Louis University. He went on to receive a PhD from Texas A&M University under the direction of David Russell. He then did postdoctoral work at the University of Cambridge under the direction of Dame Carol Robinson and was awarded the first ever Waters Research Fellowship. Brandon then moved to the University of Michigan in 2009. Where he seeks to enable breakthroughs in structural biology and drug discovery by leveraging the potential of ion mobility mass spectrometry for comprehensive three-dimensional analysis of the proteome. His team leads efforts in the role of solvation of biomolecular structure. He's pioneered collision-induced unfolding for detecting the structural state of protein-ligand complexes, and he's investigated structural consequences of binding drug-like compounds to misfolded protein targets. Professor Rutlow's work has resulted in greater than 130 peer-reviewed publications and many awards, including the Eli Lilly Award in Analytical Chemistry, the NSF Career Award, the ASMS Research Award, the Protein Science Young Investigator Award, and the Agilent Thought Leader Award. It's my pleasure to welcome Professor Rutlow. Professor Rutlow, the floor is yours.
Well, thank you, Chris, for the introduction. I'd also like to extend my thanks to the CAS organizational team for inviting me back. It's always great to be back at CAS. Of course, it'd be better if we were all in the same room, but I think this is still going to be a great venue for us to have a conversation about protein stability measurements, which is the focus of my presentation today. So before we get into some of the details of the data I want to show you, I thought we'd take some time and talk about the motivations behind the work that we're doing in my lab at the University of Michigan, especially in the context of some of our perspectives on the challenges that currently face us in the drug discovery and development. So I probably don't have to describe to this audiences the big picture challenges associated with drug discovery. It's obviously a very challenging endeavor overall. We're faced with this huge chemical space to sift through in order to take our pathway through to find FDA-approved medications, and this, of course, can take a lot of time and be very costly to complete. There's lots of reasons for this, but when one begins to look at this pipeline from the point of view of a measurement scientist, you begin to see that there are an array of different technologies positioned along this pipeline so that we can get the best information possible in order to fail drugs more quickly and move drugs through the pipeline in a way that's the most cost-effective and time-effective way possible. Of course, the beginning of this pipeline really leverages a lot of high-throughput technologies, some of which are computational in origin, that are aiming to give us very directed and specific information, one-dimensional information about the action of potential compounds against a target. The concept really of structural biology doesn't really rear its head in this pipeline until much, much later on. And that's because structural biology, as many of you know, is generally a quite expensive analytical option to take, both in the context of time and also in the context of sample. But I would argue, as we start to think about the ways that this pipeline needs to be flexible to accommodate different types of targets, perhaps those that are more coupled to disease via conformational switches than enzyme activity, one needs to think about how to put structural biology more front and center in the context of how we think about drug screening. And I would argue that this challenge becomes even more important to consider when thinking about biotherapeutic development, when the proteins involved here act as both, obviously, the targets as well as the drugs, and have their own dynamism and structural manifold to consider when thinking about their mode of action. So my group is really aiming at trying to develop immobility masterometry to assess some of these challenges. And I'm going to introduce this technology to you as well as push it forward and give you some data to think about in the context of how it's being used right now in my laboratory to address both biotherapeutics as well as some small molecule drug discovery type applications. So the first concept we have to get around is that of native masterometry, which Albert covered in his keynote yesterday. Just to remind you, the idea here is that we're going to try and develop approaches that directly capture proteins and their complexes from solution using nano-electrospray ionization and carry those as ions into the instrument for direct measurement. So this is usually accomplished by pretty small emitter geometries on the micron scale or smaller, using buffers like ammonium acetate that are designed to replace some of the ionic strength components associated with the native environment of proteins and complexes, but also are volatile enough to leave the protein during the desolvation process. So the idea is that we capture our proteins and complexes within individual droplets that are charged, and those charged droplets dry down on the millisecond timescale to give us bare ions that resemble strongly the structural states that these complexes had in solution. And there's lots of ways that we know that there's a correlation between these different environments, that of the gas phase and of the solution phase, because for example, we can capture oligomeric state populations of protein complexes that resemble strongly those that we know to exist in solution. For example, in this case, we have data for the Groyel tetradecamer, which is sort of like a standard in the native masterometry community, as well as other forms of structural biology, where we can capture just data sets that indicate the 14-membered Groyel tetradecamer type state that we know to be the active configuration for the protein in solution. And this, of course, comports with that other structural biology technologies can tell us about the overall oligomeric state populations of Groyel. And this is simply a colorimetrically indicated data set here from electron microscopy. So I would argue that native masterometry is really one of the premier methods that you can use to assess the oligomeric state of proteins and protein complexes. You can also use this technology to directly observe the ligand or drug binding in proteins and assess the stoichiometries of those types of interactions. And by using the relative intensities of both the bound and the apo states, you can also determine KD values from these types of measurements if you carefully control for different factors associated with the measurement. So my group takes this native masterometry approach and graphs it onto immobility masterometry. We use two different platforms, mainly in my laboratory, to do these types of measurements. The Agilent 6560, which my group has really helped to modify for optimal use for native masterometry, as well as the Waters Synapse G2, which we have used for a number of years now for doing high-quality native masterometry, as well as immobility separation. The basic principles involved in the way that immobility takes place in these instruments, and again, for those of you who don't know what immobility is, it's a sort of a gas phase form of electrophoresis-based separation. It takes place in the millisecond timescale, but can separate ions and proteins and complexes according to their size and charge on that timescale. The principles of mobility separation of these instrument platforms are different. One uses a drift tube type format, the other one uses a traveling wave format. The details of that are not super important. What will be important for our conversation later is that the positions of the analyzers here that I'm coloring here in green, if we can see my mouse, the immobility analyzer located here and here are at different points inside the instrument. And as we start to compare and contrast these instrument platforms throughout my presentation, that'll become important, I think, to keep in your mind. But in terms of the operational principles behind the instruments, they're somewhat similar, right? So we have a mobility separation that takes place first. We introduce ions into a ring stack, an ion guide. Inside that ion guide, we positioned a partial pressure of a neutral gas. In our case, this is usually a mixture of helium and nitrogen, or maybe just pure nitrogen. The ions are in the influence of electric field. They will drift along that electric field and collide with the background gas. The larger my ions are, the more they collide with the background gas, and the slower they travel through the device versus the more compact, streamlined forms of those ions. So they elute from this chamber on the millisecond timescale and give us some form of separation that, again, is related to the size and shape of the ions if we keep charge fixed. They then can be analyzed directly by time of flight masterometry on a faster timescale. And we can, of course, record the mass spectrum for all of the ions that elute from the mobility chamber. And we can combine these information streams together to give us a three-dimensional data projection where we were plotting drift time on the y-axis, mass charge on the x-axis, and the intensity of the ions are now shown on different, in this case, colorimetric scales. So my group has spent some time thinking about how accurately and how precisely we can measure these sizes of proteins using ion mobility type separation. So the size that you typically extract from mobility data is called the collision cross-section, or CCS for short. So we've used the Agilent 6560 to record, and just published last year, a range of proteins and complexes in terms of their cross-section, and compared these with previous measurements across some of these proteins on the same instrument platform, and found really good reproducibility between those measurements and indeed replicates on our own platform to the tune of around 1% RSD, and average values are actually a bit lower than that. When you cross-compare these 6560 cross-section measurements with those that have been taken for well over a decade on the Synapt-based platform, we see some differences. Now, these comparisons are actually for both of them drift tube-based separators, but again, the positioning of the drift tube is different. So these older data from this paper that Matt Bush and I published in 2010 was taken from a drift tube that was implanted in that same region of the instrument that I showed you on the Synapt platform, deeper inside, much after the Electrospray device, whereas our measurements from the 6560 take place much closer to the ion source. And you can see here, there's a sort of a 3% offset between the cross-sectional areas on both of these platforms, which we think is related to the different lifetimes of the ions involved in these different measurements. This is not something that you would typically see for small ions, but since proteins are so flexible and dynamic, these types of changes can be important to consider when one thinks about databasing these types of values and using them from the point of view of structure determination, or in the context of simply identifying protein isoforms associated with these different classes of protein. Another potential challenge here is that, again, proteins are very dynamic entities, and they occupy a range of structural forms in solution prior to our analyses. And just to make this particularly relevant for our conversations about biotherapeutics, here's an overlay of about 360 individual model structures for antibodies derived from IPET-type measurements. This is courtesy of Gary Wren's laboratory at Lawrence Berkeley National Laboratory. Here, what we're looking at is an overlay of all of these structures to give you a sense for what people think are the dynamic range of structures that antibodies can adopt during their motions and functions in solution. So the question is, does this ensemble of structures get transported into the gas phase, and to what extent can we probe this ensemble of structures using gas phase type mobility measurements? So if I take each of these 360 structures and measure and estimate their cross-sectional area, and then project them at different levels of resolution in terms of mobility separation, I see that at typical values of mobility separation resolution I can achieve on either the 6560 or the Synapt G2 platforms, I can see essentially a single peak for this entire ensemble. So all 360 structures, all their cross sections are encompassed in this single peak, the single distribution. As I begin to increase the resolution of my mobility separator to a factor of 6 beyond what we can achieve experimentally now, I begin to resolve additional structural states from this ensemble. If I then increase that by a factor of 2 and further another factor of 2.5 up to a complete resolution of around 600 or so, then I can resolve about 15% of that original 360 structures, which isn't great, and gives you a sense of the challenge here. Again, if we're dealing with these dynamic entities with lots of potential microstates involved in their structural ensemble, then collision cross-sections become a really challenging place to exist when one wants to pull out details of the tertiary structures of biomolecules. So because of that, my group has been pioneering this technology called collision-induced unfolding, CIU for short. The idea here is that we can take proteins sprayed from our Nanospray, native Nanospray experiment that exists in a range of different charge states. We can select one of these different charge states, observe the ground state arrival time distribution for these ions, similar way that we did on the previous slide. But we can take these ions and begin to heat them up in the ion trap that precedes the ion mobility separator in each of these instrument platforms. And if we do that, we see that the ions change their size as a function of the level of activation or ion heating that we apply in the experiment. And each of these states can be recorded and tracked with precision, giving rise to what we call CIU fingerprints, right? So the basic idea here is that the pattern of states that you record in these experiments can be diagnostic for the beginning structural states that we have in the start of the experiment. My group has developed software approaches that we can use to probe this data quantitatively to a level of precision that allows us to make, I think, really deep and meaningful conclusions about the structural states of these proteins, especially for biotherapeutics, which is going to be our focus for today. So the first vignette I wanted to talk to you about is our work with biosimilars. Here, we're going to be looking at this basic challenge of how do we take antibodies that are designed to be essentially identical to a reference or innovator product, but are prepared in different ways and maybe have slightly different formulations or maybe even slightly different post-translational modification states in terms of their glycosylations, for example. How we compare and contrast these two, especially from a higher order structure point of view. We think ion mobility Masterometry has a lot to offer there. So the examples I'm going to show you today, we're looking at a group of infliximabs, Remicade from Janssen, Inflectra from Pfizer, and Renflexis from Merck. All of these are the same basic archetype, the infliximab, which is basically a form of biotherapeutic that has had a pretty big role in the biosimilar space for the moment. I mean, it's one of the very first classes of biosimilars that have been approved by the FDA. They're all therapeutics that are associated primarily with rheumatology, Crohn's disease, for example, and they're all tumor necrosis factor or TNF-alpha type binders. So here's some example ion mobility Masterometry data collected for Remicade here as an example. Under both native and heat-stressed conditions, we can see that we get nice narrow populations associated with drift time and a nice tight distribution of charge states and a little bit of dimer for the native state of the biotherapeutic. And when we heat stress it, we get a lot more dimer as well as some fragments here, the FC region and the Fab region we can observe and resolve in our data sets. Of course, we record CIU data from all of these different states that we observe as a function of heat stress, for example. And it's hard to see in just looking at these different fingerprints, but there are actually some remarkable differences here between the native and the heat stress sample. First and foremost, we can observe that there are shifts in both the widths and the centroid cross sections for some of the features that we observe here. You can see we've boxed in three main features from these data sets. So if we probe, for example, this first feature, which appears at lower voltage here, we can observe that the native state, like I've already mentioned, is this broad distribution that probably encompasses this, grand number of structures from, similar to what, Gary Wren's models show us. And as we then heat this antibody, we can see that the structural population anneals is something much more narrow, which is interesting. But then as we begin to heat these structures, the centroids of the features that we observe also shift subtly. You can see the level of shifting going on here at this high voltage slice. So the software that we've developed can actually go in and chart what we call CIU50 values, which are the transition points in voltage space for each of the different features that we observe here. This just tracks the transition from feature 1 to feature 2, that's CIU50 number 1, and then feature 2 to feature 3 would be a CIU52. And we can see from this data that we can see for each of our biotherapeutics here, Remicade, Inflectra, and Renflexis, that the native data here across charge states allows us to differentiate these different antibodies based simply on stability, but also the stress states here, which again are indicated in these darker colors, are different still, right? So all of this then paints the picture that immobility masterometry and CIU together can track a lot of details surrounding the high-order structure of antibodies and how they shift as a function of heat stress. We also had questions about the general reproducibility of CIU data, and so we took, to date, the largest population of replicates, both across lots and across technical replicates for CIU data. And you can see a heat map plot for all those different data sets here. So each of these boxes represents an individual CIU fingerprint we collected in both native and stress states. And you can see we're comparing them across the entire fingerprint using RMSD axis to do that. And the comparison point is always Remicade here. You can see the level of difference across each of these different replicates here is pretty minor, but the difference across biosimilars here is dramatic, to the tune of factors of 2 to 5 different, depending upon which level of data you're looking at here. So we also have built machine learning algorithms that go into this type of data and pull out classifiers that you can use to identify which biotherapeutic you're looking at. And we can do this to great levels of accuracy, but our accuracy is maximized if we include data across all the chart states in the data, as well as across both native and stress states. We've also collected differential scanning calorimetry, or DSC, data across all these different antibodies, across all these different states. And we can observe, and I'm sorry this isn't labeled, but this is the native data for Remicade, reflexes, and Inflectra. And this is the stressed data over here. You can see that there's some minor differences in the CH3 melt temperature for the native states here, but all of this gets pretty much blurred out when you look at the stressed data sets here for DSC. But if you just look at individual slices from these drift time distributions that are collected from CIU data, you can see some pretty big differences associated with some of these biotherapeutics. And some of them, for example, this 135-volt slice here, you can use to parse between each of these different antibodies in both native and stress conditions. So again, the take-home message here is that CIU is able to differentiate some differences in stability that are simply too subtle to track using classical DSC type measurements. The next vignette I want to share with you is some collaborative work we're doing with AbbVie. Our contact there is Joe Eschweiler. So here the question is about the dimer structures produced upon stress for a group of antibodies. Here we're going to be stressing the antibodies a couple different ways, most notably using heat and pH, and asking questions about the different structures the dimers might take upon stress. And here are some examples of what we may see from some older TEM images showing that dimers can form sort of in a face-on configuration, or using sort of a tailed configuration, or other types of overlapping type structures. The goal here is really understand the mechanism of dimer formation and if the mode of stress has an impact on the dimer forms that are created. We're going to be looking at two different antibodies here, one termed alpha and one termed beta. They're essentially the same sequence, but they have differences in their CDR regions. We can collect all of the native Masterometry data for these types of samples on both native conditions, heat stress conditions, and pH stress conditions. And we can see oligomers all the way up to tetramer under the conditions, especially on pH stress. And we can track the differences in aggregate populations as a function of either the heat stress, we're incubating at 50 degrees C in histidine formulation, all the way up to 180 minutes, and we can see up to about 6% dimer formation under these conditions. We can see a lot more dimer formed under pH stress conditions, up to about 30% of the population. But again, that difference, that aggregation is different, appears for the beta and alpha antibodies, even though the difference here is only in the CDR. We can, of course, track the CIU data across each of these different states. These are monomer CIU data for native, the 50 degrees C, and pH3 type measurements. You can see that there's some pretty dramatic differences associated with heat stress, like we saw in infliximabs, but also in the pH stress data. And we can track those differences in terms of the overall RMSD across the entire fingerprint for each one of these different antibodies, and observe, again, there are differences between the two antibodies that we can track, as well as we can track differences that are associated with the higher order structure for both pH stress and heat stress here. Again, the end of the road here was always going to be aiming at dimer structures here. So these are dimer cross-sections recorded across both the SYNAPT G2 and the 6560 across a couple different stress conditions here. And they range from about 120 to about 135 square nanometers. We were able to develop a number of different dimer models that are created from those EM data archetypes that I showed you a few slides back. And then we ran these through some MD simulations that allowed us to compact these so that they agreed with the cross sections of the gas phase measurements that we're taking here. And we observed that the range of experimental data really led us to two different structural populations and allowed us to eliminate all of these other much more elongated dimer populations from consideration. So it seems like under most conditions, we're forming pretty compact dimers that best agree with this complete overlap type model, where all of the faces of the antibodies are basically in contact with each other. But under pH stress conditions, especially, we think we're forming potentially a population that skews towards this inversed overlapped type structure, where basically the axes between the two different antibodies are inverted. So the last biotherapeutic vignette I want to show you is our work that we're doing on A-beta binding antibodies. This is relatively recent stuff that we're doing in collaboration with Pete Tessier's lab here at the University of Michigan. So we're targeting a bunch of different antibodies in this space, but our inspiration was definitely aducanumab, the famous antibody that was just approved over the summer for targeting A-beta. These are, of course, really interesting and potentially controversial biotherapeutics for lots of reasons. But there's a clear analytical challenge here that we feel that A-beta itself is a pretty complex molecule that occupies lots of different oligomeric states. And our impression is that current methods that are used to assay these types of complexes are lacking in terms of resolution. They mostly rely upon page-tail type analyses of the different oligomers that we're tracking and how they bind to the A-beta antibody. And of course, there's a lot of other types of antibodies in this space that are under development. Here's a list here at the bottom of the slide. So we feel like developing native Masterometry and Ionization Masterometry tools that can assess these types of problems might be really useful for the field to consider as we move forward with these antibodies in the future. So what we're doing here is we're taking three different antibodies here as a starting point: aducanumab, obviously, crenezumab, which is a Genentech product associated with the same type of idea, and A34, which is an antibody developed by Pete's lab here at UM, where they're trying to mimic aducanumab but improve it slightly, basically. So we're challenging these antibodies with three different A-beta proteoforms: A-beta 1 through 40, 1 through 42, and A-beta 3 through 40 in concentrations, 30 micromolar and 100 micromolar. And the idea here is that we're going to pre-aggregate both of these populations, but the higher concentration is going to concentrate the sample for larger amounts of oligomers that they can use. And we're going to track both the native immobility Masterometry data as well as the CIU data. So here's what some of the data look like. So this is just for the antibodies on their own. Aducanumab, crenezumab generate charge states, native charge states that you might expect. A34's got some satellite peaks here that relate to different isoforms, truncations of the sequence that we can also remove from our analysis if we want to, but it appears to be, just as structurally stable as the other antibodies in the screen. We challenge this with A-beta 1 through 40 at 30 micromolar. We see that we can observe direct binding of A-beta to the antibody, and observe that by mass shifts associated with the antibody cluster we were tracking here. For aducanumab, we can see both 1-to-1 and 1-to-2 stoichiometries. For A34, we can see 1-to-1 and 1-to-2 stoichiometries as well. But for crenezumab, we observe just exclusively a 1-to-2 stoichiometry. If we take a look now at the 3 through 40 isoform here, we don't see any binding at all for aducanumab and no binding here for crenezumab, but we see the retained 1-to-2 stoichiometry for crenezumab. And that's expected because the known epitope for aducanumab and A34, because it's based on aducanumab, is the N-terminus of A-beta. So it seems like our methodology here is working as expected. I want to pause here and briefly sort of point out that under these conditions where we're observing these complex binding morphologies here in this region of the spectrum where we've been focusing on for the last two slides, we can also track just the free oligomer populations of the A beta at lower mass charge, and we can see evidence for monomers, dimers, trimers, and tetramers in that low mass charge portion of the spectrum at the conditions that we're showing you here. So if I summarize all these results, basically for aducanumab, we can see evidence of up to 1 to 2 stoichiometries, and we can see the same thing for A34. Crenezumab seems to bind 1 to 2 stoichiometries exclusively. At low concentrations, we pump these up to high concentrations. There's really no evidence that aducanumab binds anything beyond this 1 to 2 stoichiometry, but A34 does show evidence of binding up to what could be tetramer A beta. And crenezumab also examples of binding, higher order binding up to 1 to 3 stoichiometries. So we took CIU data of all of these to track the stability changes associated with each of these bound states, and we could observe clear differences associated with each of these antibodies, indicating that the binding events here increase the stability of the structure to different extents. Aducanumab has, again, stability increases as you might expect for its 1 to 1 and 1 to 2 binding. That slope, if you will, in stability increase is shallower for crenezumab. We don't see as much of a stability increase for binding either the 1 to 2 stoichiometries or 1 to 3 stoichiometries that we can observe. And the steepest slope here is for this A34 species, which goes all the way up to this level of stability up here for this 1 to 4 type stoichiometry that we observe. All right, so the last vignette I want to show you is really based around our recent results that are focusing on making a CIU screening-based methodology. And so for this experiment, we're focusing on some small molecule type screening applications. The key challenge here, of course, is to come up with a methodology that we can use to introduce samples very, very quickly to the instrument. And one of the ways that we're doing this is working with Bob Kennedy's laboratory here at the University of Michigan. Bob's group is experts in developing these microdroplet microreactor type structures. So the idea here is that we can take a multi-well plate and take aqueous samples and overlay a layer of perfluorinated oil on top of these. We then come along with a tube that's connected to a syringe pump and simply aspirate each of these wells into this tube, and that creates now a segmented flow by which aqueous droplets are segmented from each other, from each of these wells, using perfluorinated oil. We then take this archived droplet-based tube, connect that directly to our home-built transfer union for electrospray ionization, and we perform our native ionizability masterometry data directly from each of these now encapsulated droplets. So the data looks something like this, and this gives you a sense for the amount of time we're taking for each of these analyses, right? So each of our sample droplets comes out basically on the time scale of about 20 to 30 seconds or so, maybe a little bit less. Then we have wash droplets that intersperse each of these different sample droplets. So the data I'll show you on subsequent slides are really from multiple of these sample droplets, but each one of these basically encompasses a few seconds worth of data collection time. So, but from that, we can extract directly native masterometry data. So the example I'm going to show you is for a sirtuin, SIRT5 here. This is just an example binding to a compound called 1066, which is a model compound that we're using here to illustrate the potential information content here. It's a known inhibitor of SIRT5. But basically, we can observe now through these clusters, we can observe both APO data as well as 1 to 1 and 1 to 2 geometries of the inhibitor bound to the protein. From each one of these individual states that we can observe, we can collect complete CIU data in the same time frame. So we can track, it looks like one transition and a second transition here. We can use our software to automatically track the stability shifts associated with each of these different transitions that are influenced by ligand binding. And from this, we can tell you how much this ligand, and this is now for a different ligand here, but it's the same basic concept, how much these ligands are shifting the overall stability of the protein upon each of these different transitions. So I think the key here is that we can get complete CIU data recorded in between 50 to 30 seconds, and the reproducibility between these CIU data that we can collect across each of these droplets is on the order of about 1% root mean square deviation. So we took basically a 96 compound screen as our first test here for Cert 5. And again, this is now data where we're tracking this first CIU transition on this y-axis against the compound library. You can see some of these transitions, the vast majority of them indicate no change from APO. That's what this green area represents, a plus or minus 3 sigma value for the CIU response for APO. Beyond that would be what we would call either a quantitative transition that leads to a stability increase for most of these compounds up here. And for down here, we're seeing a couple compounds that actually destabilize the protein. The second transition here looks like most of these compounds end up stabilizing the protein for the second structural transition. If we were to try and take those known inhibitors out of this population, and note the number of hits that we have here is large, but that's because we've packed this compound library with analogs of known inhibitors. So we're expecting to see lots and lots of hits. But for those inhibitors that are known and have known IC50 values, we can actually create a correlation now between the CIU50 data. And we can see pretty good correlation with one outlier here for CIU51, and CIU52 here, we're seeing a negative correlation here, which has a much better correlation coefficient. Another key, I think, element here to focus on is this entire 96 compound screen with CIU was accomplished in 60 minutes. This includes 10 seconds per sample, which were performed in triplicate. So we took 3 droplets of each, and they also included the watch steps in between the samples, right? So we were actually able to develop an automated approach analyzing all this data using our software, which again goes in and uses machine learning and looks for stability increases associated with known inhibitors and tries to map those onto the rest of the data set. And we could use this pretty routinely now to go in and classify those compounds that look like APO versus those that look like they are stabilizing the protein, basically. And so that's what we're picking out here. And a couple of these gray points are some of our controls here that indicate those that we're expecting to see in this pattern, and those align with the rest of these compounds as expected. Okay, so that's all I have to show you today. Looks like I'm a little bit ahead of schedule. So I want to conclude by saying that I hope I've given you a sort of a tour through structural masterometry in terms of this growing area of masterometry and what it can do for drug discovery and development. CIU, I feel, is a rapid and potentially pretty transformative technology for doing stability assessments that really only requires a very small amount of sample and doesn't require a purified sample or the need for labeling in order to get the stability type data. And our data here for biotherapeutics and small molecule drugs really highlight, I feel, the level of information richness we can achieve with this type of analysis and the potential of generating the data very, very quickly in a screening type mode. All right, so I want to acknowledge the folks that did the work here. So Dr. Varun Gadarki really pioneered these comparisons between the 6560 and the Synapse G2 instrument that I showed you early on in this presentation, the paper we published this last year. Kara D'Amico, who's a joint student between my laboratory and Bob Kennedy's laboratory, did all of those CIU screening type measurements I showed you at the end with Cert 5. Dan Vallejo, who recently graduated and is now a postdoc in Facundo Fernandez's lab, did all of the biosimilar analyses that I showed you, as well as led that AbbVie project I showed you. And Yilin Han, who's a current student in the lab, but is now currently on a internship with Biogen. She did the A-beta binding antibody type measurements that I showed you. Of course, our collaborators here from AbbVie, Joe Eschweiler, Agilent Technologies, including folks like Brian Miller, John Felstad, Ruan Kuagama, and George Stafford, have been key partners in developing the 6560 for these types of measurements. Our University of Michigan collaborators include the Kennedy Lab for those Saturn-5 type screening measurements, and the Schwendemann Lab, who's been our partner for those infliximab type biosimilar measurements that I showed you. Of course, our funding sources and you for your attention. Thanks so much for inviting me, and I look forward to the Q&A.
Great. Thank you, Professor Rutlow, for the excellent keynote presentation. It's very interesting to see how far the CIU has gone and how practical it can be used for the pharmaceutical industry. So we're going to move to the Q&A session. I will start with the first question. Have you seen any challenges with GrowEL purity impacting your analysis?
GrowEL purity. So if you're talking about GrowEL sourced from, say, for example, Sigma, then yes, that can be a problem. But there are protocols you can get in a number of different publications to purify GrowEL from that source to create it in a form that you would use for standard measurements by native Masterometry or native ionizability Masterometry. And if you're interested, I can provide the references for that protocol. Basically, it involves denaturing GrowEL, going through a couple of cleaning steps, and then reassembling the GrowEL in presence of magnesium and ATP at the end of the protocol. And then usually following that would be a final exclusion type purification step. And that's the protocol that we would use to take GrowEL from a commercial source and use it as a standard. But of course, if you're expressing and purifying it on your own, then you wouldn't have to worry about those sorts of things probably.
Great, thank you.
The next question we have is, could you comment on how you ensure dimer formation of MABs is not a source artifact during the ionization process?
Yeah, that's a great question. So we've learned a lot over the years about how to track source artifacts, oligomers that form via electrospray versus those that are present in solution. And most of that result revolves around concentration-dependent measurements. So most of the measurements that you saw today for those dimer measurements were taken at very, very low concentration to make sure that that type of process wasn't happening. So we're talking like less than 5 micromolar, basically. But you can even go lower than that and still see a lot of these dimers populate. The other, I think, key here is that for a lot of our measurements, they were taken as a function of some sort of stress condition, for example, heat or pH. And if you see the dimers grow in as a function of those stresses, that you also can relate those directly to solution phase dimer populations, because of course, electrospray in those conditions are a constant during all of the measurements.
Great, thank you.
Great. Next question. For infliximab sample, can you describe why you observe a higher intensity of the dimer species under heat stress condition versus native condition? Was this expected for the sample?
Yeah, so in our experience, heat stress usually produces a certain amount of dimer, and we've also measured that by solution separations as well, which is a more standard approach for measuring dimer concentrations. And heat stresses, typically with infliximabs, it depends on you do the heat stress, but you can easily generate, 4 to 6% dimer under those conditions. And so that wasn't a surprise to us, no.
Okay, thank you.
So our next question is it possible to determine which AB oligomers are preferentially bound by antibodies? Is there a change in the leftover oligomer populations after adding the antibodies?
Those are two really good questions. So the answer to the first question, I think, is yes, but we haven't gotten there yet. So what I showed you was all, presented in the form of stoichiometry of binding, because we don't know if those, say, for example, a 1-to-2 stoichiometry is simply 1-to-1 binding on each arm of the fab, or it's in fact a dimer binding to one arm of the fab. So in cases where we see higher order complexes, 1-to-3 and 1-to-4, I think that those are likely evidence of oligomeric binding, unless there's some other low affinity site elsewhere not indicated on the fab arm that is capable of binding A beta, but I think that's super unlikely. So what we're trying to do in that context is work through some other types of data that might help us decouple the idea of 1-to-1 binding on either arm versus a dimer binding event. And the tool that we're turning to mostly there is HD exchange Masterometry to map the locations of antigen binding at each of those concentrations that I'm showing you. So I think that's possible, right? And I think that we'll probably get there before we end up finishing that project. But in the context of your second question, how does the A beta population, how is that altered when we add the antibody in? We don't see much in the way of alterations, but to be honest, we haven't looked in detail at that question. I think that'd be interesting to track in the future.
Thank you.
All right. Next question. How does collision-induced unfolding in protein correlate with the structure differences in solution? I guess the question is asked the gas phase protein structure related to the solution phase. That's the first question. Second, how to explain the CIU differences for different charge states?
Yeah. All right. So the first question is quite broad, right? And I would say there are lots of elements of general native structure that we think are preserved in the gas phase. And what I mean by that is that if you take the measurements in the right way, in other words, you don't add too much energy to the ions and you measure them quite quickly, elements of overall quaternary structure can be retained, some elements of tertiary structure and secondary structure can be maintained. What we think are lost is, elements of position of residues on the surface. There's lots of evidence suggesting that collapse that occurs in the gas phase for structure mostly occurs at the surface of the biomolecules. But I think, there's different answers to that question based on different classes of biomolecule. For example, antibodies specifically are known to collapse quite a lot in the gas phase, more than average. And most of that collapse is thought to occur around the hinge region of the antibody. So when we talk about native structures in the gas phase, what we're really saying is that there's a native-like population that retains as much as possible of the configurations of the biomolecules in solution. When it comes to the second part of the question was related to CIU data, is that right?
Yeah, the CIU kind of discrepancy between different charge states.
Yeah, so that is something that we've observed in the past as well, really all the way back to the beginning of our experiments with CIU. And it relates to the mechanism by which proteins unfold, right? So when you unfold a gas phase ion, it has all of this charge decorating its surface. As you begin to release more surface area, charge migration occurs, and that charge migration can accelerate the unfolding process. And obviously, if you have more charge on your surface, you get more of that. The key thing to do, I think, in comparing CIU data is to make sure that charge is a fixed value. It's not a variable in the context of the experiment. So if you compare across antibodies, for example, or across replicates, you're always comparing the same charge state there to make sure that those charge-based differences are not tripping you up. But in many cases, we would opt to look at the lowest possible charge states in our experiments in order to minimize any charge-based aberration of structure. But in some cases, we actually prefer to look at higher charge states because the more charge you place in the biomolecule, the more transitions you might see typically. And the more transitions you see, the more unique the fingerprint becomes, basically. So if you're looking at just fingerprinting differences, higher charge states might actually be better than lower charge states. But that's part of the metric that we use. And like I showed you in my presentation, a lot of what we do most recently when we're building like machine learning based classifiers is just take all the data across all charge states and treat it all as a potentially differentiating population.
Yeah, great.
So the next question we have is, how does the 10-second CIU compare to the typical CIU collected for minutes? Is there any loss of resolution, unfolding features? Do you see any challenges in applying this CIU-based screening approach to larger protein complex targets?
Okay, so the answer to that question is entirely based on signal intensity, right? So if you have a well-spraying sample that produces lots of signal, then the differences that you see between a 10-second CIU and a 2-minute CIU are minimal, I would think. If you have a doesn't spray super well, doesn't produce a lot of signal, then you might see some differences. You might lose features, you might lose intensity, you might lose the ability to track transitions with high accuracy and fidelity. But in the case that I showed you, all those samples were sprayed very, very well. So 10 seconds, that data matched what we were able to collect at higher integration times without any sort of loss of information content or anything like that. Expanding that concept to bigger protein complexes is definitely something we're interested in doing, right? And we've started doing that already. We're really interested in trying to leverage that concept for protein-protein interaction inhibitor type screening. We're looking at a protein complex and looking at how an inhibitor can come in and disrupt the assembly. It's a big picture challenge right now to find technologies that can get after that problem in a way that's pretty universal, and it's something that I think that high throughput CIU could help you do, because if you see an inhibitor binding event, the CIU could tell you about maybe which domain in the protein you're hitting and to what extent and to what the mechanism might be in terms of stabilizing the protein structure or destabilizing it in a specific way that it might help you in the context of pushing ligands forward that might do better at that sort of PPI type mode. So I think, yeah, we've thought definitely about doing it, and it's early days yet, but I think it's certainly possible to do, and it probably really powerful to do as well.
Thank you.
Cool. We'll move on to the next question. I think you addressed the difference between proving gas phase and solution phase. So we'll go to the next question, which is, do you observe differences in Ciu fingerprints for the same sample in different mobility platforms? For example, DT versus TWIMS.
Yeah, so we do observe some differences. They mostly manifest as differences in the cross-sectional areas of the features that we record, and some differences between the transition voltages. And it's really hard to compare transition voltages between the two platforms mentioned there, because they exist in different pressure regimes and different voltage regimes. So cross-comparing is very, very challenging. But what doesn't change so much is the number of features that we observe and their ability to do work in the context of differentiating different isoforms. So they tend to have the same abilities in the context of like pulling apart antibody isoforms or ligand binding events, and they tend to do it in the same sort of ways. Like the same features that move in one data set would be the same features that sort of move around in the other feature in the other data set in the context of what's giving the information content to make, assessment about structure and so forth. And in some cases, the offsets in terms of cross sections are systematic, right? So like I mentioned, on the 6560, a drift tube instrument, cross-sectional areas are systematically a little bit lower than those on the traveling wave-based mobility separator type data that you typically use for calibration. The drift tube data sort of originated that. Anyway, those two data are offset, and it turns out that the cross-sectional areas, as if they're unfolded states, are similarly offset in many cases. But I would say if you look at them, there's more similarities than there are differences in most cases, right? And I think that is heartening to us, because we're really interested in trying to think about databasing CIU data that you could use and refer to as a reference point to identify protein isoforms or different potential sort of like reference molecules, if you will, if you're trying to do something high throughput and screen. So I think that those types of cross-platform comparisons give us, I think, a lot of room to be optimistic about how useful that might be in the future.
Yeah. Extending to the question, do you think it's possible to use a standard to calibrate both systems to bridge data together?
Yeah, so we've tried to do a little bit of that. And I would say that initial efforts there, where we thought it would be pretty straightforward to do cross-calibration, turned out to be less so. So we've actually taken a step back there, and we're collaborating with Jim Prell's group at University of Oregon to actually understand more of the ion physics and heating that's going on in the drift tube experiment, so that we can better compare it to the SYNAPT G2, for which Jim has already developed a model that describes the internal temperature of the ions. And so I think what's probably going to bear fruit is making sure we understand the internal temperatures from a theory point of view first, before we start to do calibrative type cross comparisons there.
Great.
So the next question is a different perspective of the previous question. Do you see differences in Ciu fingerprints or Ciu 50 values with differences in desolvation that may come from different nano ESI tips?
Yeah, so not so much from different tips, but we've definitely seen differences from different buffers. We've also played around with like formulation screening using Ciu, and you can definitely see differences associated with formulation elements sort of adhering to proteins during the electrospray, and you can see shifts in stability associated with them. And I think that's actually a growth area for Ciu personally, is the idea of trying to use it as a means of trying to better sort of pick formulation additives for altering stability. of biotherapeutics. But yeah, so in terms of like it adding like a layer of noise on CIU data, in our experience, if you keep formulations simple and buffers simple, that's not so much of a problem. The other thing that you can play around with, which we started to do, is using nanoscale electrospray emitters. So this is a concept, described by folks like Evan Williams, for example, where you can take nanospray tips, then usually accommodate sort of a micron scale exit aperture, and you can shrink that down to the nanometer scale. And that gives you a lot more salt tolerance and tends to give you less adducts as a result of that. And in those cases, you can pump the concentrations up of different formulation components and these sorts of things while still maintaining pretty clean looking spectrum. So yeah, I think for really high concentrations of salts in solution, it's still an issue, definitely. But for modest concentrations of salts, like sort of like in the, certainly, micromolar to maybe low millimolar range for some salts, you can get away with it under some conditions, and you can still get pretty clean spectra, and you can get reproducible CIU data across those types of samples usually without a problem.
Great, thank you. Maybe as a follow-up question to the formulation details you brought up, do you see that this technology could become a platform approach to candidate selection?
Yeah, we would love to see it move in that direction, to be honest with you. So as part of our partnership with Agilent, we have in the lab here a Rapid Fire 400, which is, you don't know that instrument platform. It's one of these devices that you can use to address a multi-well plate with a robot, and then aspirate those samples and directly introduce those into the instrument for rapid analysis. And what we want to do is marshal that type of platform for doing experiments that look like candidate selection, basically, where we take a whole panel of sequence variants or expression variants, and we would roll through those with a plate, and we would use the CIU experiment and ionizability Masterometry experiment to assess the fitness effectively of those candidates as one dimension of a maybe multidimensional analysis for doing candidate selection.
Thank you.
All right, next question. Can you relate CIU transitions to the stability of the secondary structure in proteins?
Yeah, so in many cases, yes. I think a lot of what's bound up in that question is knowing what region of proteins are undergoing unfolding during those different transitions that I show you in the fingerprints. And for many cases, we do know that, mostly in model systems. We're still learning a bit about how to interpret that type of data for antibodies, for example. But most of our data points to the fact that the very first transition that you observe in those antibody data sets is probably related to the Fab undergoing some amount of unfolding, and later transitions relate to the FC undergoing unfolding. And we're still working on trying to get more granularity in the context of what that can tell us about different regions of the antibody and how their stability is influenced by different types of modifications or stress states. In other cases, we've mapped this much more specifically. Like we published a paper now 4 years ago where we actually used a series of truncated versions and chemical probe type bound structures of albumin to figure out the specific unfolding pathway for that particular protein. And we can now, I think, with a combination of CIU and electron capture association, which we also have in the lab as part of our collaboration with Agilent, we can actually map that much more specifically. And we think now that we can link albumin unfolding down to individual peptides that are undergoing on the unfolding transition first, based on their ability to be fragmented by ECD. So I think that's coming, probably for antibodies as well. But it's going to be some time before we get all the data together and are able to analyze it completely.
Great, thanks.
So I had one more question. Do you think that you could take CIU information and use AI to come up with a set of rules for protein behaviors that could be sort of generally applied?
Yeah, possibly. So the classification architecture that we use for CIU data, that I showed one example of that where it was that 2-color box where you're separating out different proteins. That's based on machine learning and a form of artificial intelligence. But we've not thought about holistically sort of capturing CIU data, ionizability data, native Masterometry data, and maybe other types of data forms piling all of that into some sort of AI manifold, and then using that as some, rubric for figuring out, for example, how you would better do candidate selection or other types of sort of like big picture challenges in the context of biotherapeutic discovery. It's interesting to think about, definitely, in terms of how you would select which types of data to sort of stream into the AI, and how you would then maybe even build in other technologies into that, and create some sort of multi-attribute monitoring sort of platform that all is connected to an artificial intelligence approach for sort of making the selections. I think it's a powerful idea, definitely, and I can imagine that's coming about in the future. Yeah, I think what would be needed is really sort of like partnerships that would allow us to get access to enough of a large enough screening pool to sort of like test out those ideas. But I think it's really cool to think about.
Thank you.
I think we're reaching the last question, but I do have a couple questions, if you don't mind.
Okay. Yeah, sure.
I was very excited to see the CIU can separate dimer, trimer, and tetramer, right? So, and you can even quantify that. My question is, have you compared with the conventional orthogonal SEC-AUC method to see if they agree with each other?
Yeah, we've in many experiments compared with SEC data, and we very often see very, very similar amounts of oligomer in both of these measurements. And like I said, you have to take the masterometry measurement very, very carefully in order to get it in a comparable state. Like looking at low concentrations is very important for doing it in the right way. But if you do it, I think the correlations between SEC and immobility masterometry are very, very strong, typically for quantifying oligomers. I've not done so many correlations with AUC, I think, but my guess is that would be a similar story there. So yeah, I think that this form of masterometry has got a lot of growth potential, really, when you think about quantifying oligomeric populations in antibody stress states, formulations, and so on.
Yeah, that's an area my spec never touched before. It's great. Mm-hmm. So also I'm intrigued about the mechanism behind it. In the cartoons, you show the FC region in the, after unfolding, it's kind of open up. So I'm curious, what is the energy level, right? Assume it is the kinetic energy during the collision got transferred into the internal energy and maybe into vibrational energy. And then is it strong enough to break the disulfide bond, or it just form a kind of loose structure inside the antibody?
Yeah, so we don't think that we're breaking disulfides bonds during CIU, or if we are, we're doing it very rarely, because in many cases we can see evidence, like if you see disulfide bonding variants and you can validate them in some way and then measure their CIU, those disulfide bonding variants have very, very different CIU patterns. And in many cases, we can track the mass very, very precisely across those experiments, and they don't change, right? So I think that it's unlikely that we're breaking disulfide bonds. We're probing that a lot of different ways at the moment to make sure that's not the case. So yeah, I think the models here, I think, are very early stages, very cartoon, as you pointed out. I think that, like I've already mentioned, the first transition we think is related to Fab. FC is probably later on, and it takes more energy to sort of unravel. But the mode that that's accessing and the amount of internal temperature that takes and the extent to which that's actually modifying the structure and what that form that takes, I think all of that's still sort of unknown. And we're working on that problem really with a lot of different techniques and technologies, including molecular modeling, to try and figure that out.
Yep. Thank you.